So my name is Rebecca Wright. I am an undergraduate biology student at California University of Pennsylvania. My advisor for this project was Dr. Sarah Meese, and she is an associate professor in the biology department at California University of Pennsylvania. And today I am going to be talking about the effects of the environment and the native soil microflora on the medicinal properties of Echinacea species. Echinacea is a group of perennial flowers that are native to North America, and it's commonly known as purple coneflower. There are 9 known species, but seen here are Echinacea purpurea, Echinacea angustifolia, and Echinacea pallida, which are the 3 most common species used medicinally. Echinacea has historically been used variably for medicinal reasons, most commonly in treating the common cold and flu, with a strong emphasis on upper respiratory infections. The most prominent modern use for it is for its immunostimulatory properties. There are many active compounds in Echinacea, and it exhibits a variety of secondary metabolites in its extracts. So it should definitely be noted that the concentrations and the contents of the extracts vary based upon the part of the plant used, which would be the roots or the aerial parts, including the leaves, the stems, and the flowers. And it also could vary based upon the extraction method. The contents of the extracts could also vary based on certain intrinsic and extrinsic factors, which would include the genetics, geographical location, climate, growth conditions, seasonal variation, and the time of harvest. So shown here, we see caffeic acid and its derivatives. And so this is one of the most common substances in ethanolic extracts of Echinacea. And it is known for being a mediator of the inflammatory response. So these different derivatives and caffeic acid are seen in different concentrations and different amounts based on the plant as well. So whatever species you're using, we see more of one derivative versus more of another derivative. There are many other secondary metabolites in Echinacea. So up in the top left here, we see flavonoids, which are known for the antioxidant activity in response to biostress, such as bacterial infection of the plants. We see here polysaccharides, and plant polysaccharides are a natural source of polysaccharides. And so they are studied for their activity in immunostimulation and regulation regarding anti-tumor, antiviral, anti-inflammatory, and anti-radiation activity. They're specifically known to activate and enhance macrophage activity, as well as enhancing chemical cell signaling by increasing the activity of cytokines and chemokines. Down at the bottom, we see polyacetylenes, and they are known for their cytotoxic, antimicrobial, anti-inflammatory, neurotoxic, and phototoxic activity. Lastly, we have alkyl-amides, which are not pictured here because it's a very basic backbone structure, but they are known for their immunostimulatory properties, and they do so by enhancing phagocytotic activity as well as T cell activation. And they're also active in antimicrobial, antiviral, antioxidant, anti-inflammatory, and several other properties. Echinacea is modernly used as an immune support supplement, and so you can get it in any of your local drugstores or pharmacies. It's not a prescription drug, it's just an over-the-counter drug, but it is used commonly in naturopathic and holistic healing practices because the immunostimulatory properties make it very widely used across many cultures. So, it's commonly used by many people who just want that extra immune support. So what we see in the literature is that there are many studies showing that there's absolute inhibition of bacterial growth, there's no inhibition, there's ambiguous results, there's support for helping with colds, helping with immune support. And so here we have a few examples. In the first box, you can see some studies in which Echinacea was found to be ineffective in treating the common cold. But contrasting that, there was a study in which it was effective for treating a common cold caused by coronaviruses. In the middle box, there's a study that ethanolic extracts were used of Echinacea purpurea and Echinacea angustifolia against several common human pathogens, and the results in and of the study were ambiguous. So again, some bacteria worked, in some settings it worked, In some bacteria, in some settings, it did not show any inhibition whatsoever. In this last box on the right, there's a study that shows ethanolic extractions of Echinacea angustifolia to be ineffective in inhibiting the growth of bacteria, whereas another study found that ethanolic extractions of Echinacea purpurea developed significant zones of inhibition for various bacteria. So as you can see, there are many studies out there. These are just a few samples, but the results are very ambiguous. There's many contrasting results, and some sources suggest that Echinacea is this really great supplement and this really great compound that can be used to fight various ailments. And some sources suggest maybe it really just doesn't. So with all of these inconsistencies in the literature, we developed the question, what part does the environmental microbiome play in this? So what we did was we tested the antimicrobial activity of Echinacea purpurea, and we did so by evaluating the plant from several conditions with the idea that different growth conditions would yield different results based on the varying microflora of those growth conditions. We are hypothesizing that there is some interaction taking place between the microbiome and the rhizosphere and the plant, which can be tested by evaluating the properties of the secondary metabolites. So for our experimental setup, we had samples from 2 different data sets. And so the first data set that we had was we used natural growth leaves. And so we made ethanolic extractions using a 10% ethanol solution and these natural growth leaves, which were grown in Western Pennsylvania. And so we had 3 different plants that we took leaves from. One of the plants had been growing in a yard for greater than a year. One of the plants had been growing in the yard for less than a year, and one of the plants was potted. And so we took those leaves from those 3 plants and we made the solutions and heated half of them to 40 degrees Celsius for 20 minutes and the other half to 80 degrees Celsius for 20 minutes. The second set of data that we collected was from samples that were taken from the Echinacea supplements that were seen in the earlier slide. So these were pure Echinacea supplements. We got them from Rite. And so we made solutions of 76 milligrams per milliliter, 38 milligrams per milliliter, 19 milligrams per milliliter, 7.6, 3.8, and 1.9 milligrams per milliliter. And so how we made those solutions was we mixed the capsule contents, so not the capsule, but just the inside contents, which was pure Echinacea, with distilled water. And we made the 76, 38, and 19 concentrations, and then did a dilution to get the 10th of that. So with all of these samples, what we did was we used the Kirby-Bauer method of disc diffusion to test opportunistic human pathogens E. coli and Staph epidermidis against these various extractions that we had made. So what we ended up seeing, here's our first set of results. And so this is the Western Pennsylvania grown Echinacea that was tested against the Escherichia coli. And so interestingly, what we see here is that there is growth densely surrounding the discs. And so this supports the idea that there's an interaction happening between the metabolites of the Echinacea and of the bacteria. Because E. coli is part of our gut flora, it is possible that by enhancing the growth of the E. coli, Echinacea is actually supporting the immune system by strengthening that innate immunity. So E. coli lives in our gut and it's going to fight off these new pathogens that come in that don't normally live there. And if the natural gut flora is stronger, then that is going to increase the strength of the innate immunity. In contrast to that, whenever we saw the results from the Western PA grown Echinacea with the Staph epidermidis, what we saw was that there was no evident interaction happening whatsoever. We had full lawn growth across all of the plates. So it wasn't specifically around the discs. There were no zones of inhibition, and these were for all of the samples. And so this further confirms the idea that the E. coli, that is a bacteria that lives in the gut, whereas Staph epi does not. So that further confirms the idea that the immune system is being strengthened. Okay, so next we looked at the results from the pure Echinacea supplement from Rite with the E. coli. And you can see a very slight zone of inhibition on the 38 and 76 milligram per milliliter plates. And the same thing with the pure Echinacea supplement from Rite with the Staph Epi is there's a very slight zone of inhibition on the 38 and 76 milligram per mil plates. So in conclusion, there was little to no antibiotic susceptibility of E. coli or Staph epidermidis to the Echinacea purpurea extracts, except in the very highest concentrations of the pure Echinacea. There was a clear interaction between the Western PA grown Echinacea purpurea and the E. coli that suggests a potential growth stimulant was present in the Echinacea extract. So this, once again, provides support for the idea that Echinacea holds immunostimulatory properties. We had hypothesized that the environment plays a role in the ability for Echinacea to produce medicinal compounds, and that the environment and growth conditions will result in different secondary metabolites. And that was confirmed because what we saw was the natural growth leaves, the Western PA growth leaves, were exhibiting, in the case of the E. coli, a growth stimulant right around those discs. In an clear interaction, the Staph Epi had no interaction from the natural growth leaves. And that was different whenever we looked at the pure Echinacea supplement from Rite, which that growth condition would be, it was probably grown in a greenhouse in a very sterile environment. There was nothing additional in the soil, none of that natural soil contents to be able to promote any type of secondary metabolite production. And so what we saw with that is those very slight zones of inhibition around the high concentrations of Echinacea. And so once again, that absolutely confirms that there was a difference in the growth conditions as to how they reacted with the bacteria. Thank you very much for listening to my presentation. And at this time, I will be accepting any questions.